Slăvit să fie Domnul și în seara aceasta mulțumim că suntem în casa Lui și ne rugăm ca Dumnezeu să folosească aceste cuvinte spre slava Lui. Aș vrea să ne ridicăm în picioare, să citim un scurt text din Romani, capitolul 1, și vom citi de la versetul 19 la 22. Romani 1, de la 19 la 22. Și cuvântul lui Dumnezeu spune astfel: fiindcă ce se poate cunoaște despre Dumnezeu le este descoperit în ei, căci le-a fost arătat de Dumnezeu. În adevăr, însușirile nevăzute ale Lui, puterea Lui veșnică și dumnezeirea Lui se văd lămurit, clar, de la facerea lumii, când te uiți cu băgare de seamă la ele, în lucrurile făcute de El. Așa că nu se pot dezvinovăți, fiindcă măcar că au cunoscut pe Dumnezeu, nu L-au proslăvit ca Dumnezeu, nici nu i-au mulțumit, ci s-au dedat la gândiri deșarte și inima lor fără pricepere s-a întunecat. S-au fălit că sunt înțelepți și au nebunit. Amin. Să luăm locul. Mă bucur că suntem la a doua sesiune pe tema creație-evoluție. Aceasta e o temă, cum am spus și data trecută, cu care tinerii se confruntă și de aceea este appropriate ca aceste mesaje să fie în seara tinerilor. A fost o dilemă pentru mine în ce limbă să vorbesc. Știu că tinerii înțeleg engleza mult mai bine ca românește. Și știu că mulți dintre cei care sunteți aici și v-ați născut în România înțelegeți engleză. Știu că sunt și unii care nu știți engleză. Dar în seara aceasta, și cum m-am gândit că va fi mai bine cu mesajele acestea, cred că mesajele vor fi în engleză, pe aici, pe acolo, câte un pic în românește, și în fiecare săptămână, cum o să fac și azi o să fie un pic mai mult, o să fac a short review, un scurt rezumat la ce a fost data trecută. Rezumatul va fi în engleză. În seara aceasta în românește. În seara aceasta rezumatul va fi în engleză pentru cei care n-au înțeles data trecută că a fost în românește. Este o dilemă cu care ne confruntăm, fiind în țara în care suntem, și întotdeauna dilemele acestea cer și un sacrificiu. Ați venit mulți și mi-ați spus că a fost prea multă informație. Unii au zis: mulțumesc, eu aș fi putut să mai stau încă o oră, că a fost așa fain. Știu și înțeleg și lucrul ăsta. Pentru unul care a studiat în domeniu, totul pare interesant și vrei mai multe detalii. Pentru unul care poate nu a văzut niciodată așa ceva în viața lui, e clar că e mai greu să înțeleagă. Însă călătorim prin lumea aceasta, ne uităm cu trenul nostru, ne uităm pe fereastră și vedem câte ceva. Acum a venit momentul să ne uităm pe fereastră în care nu ne-am uitat poate niciodată. Așa că să nu fiți frapați dacă ceva este nou și necunoscut și poate de neînțeles. Aici, în versetul acesta, spune că Dumnezeu li se arată în mod lămurit, adică clar, oamenilor, atunci când oamenii se uită cu băgare de seamă, chiar cu microscopul electronic, la lucrurile făcute de El. Și atunci vor vedea însușirile nevăzute ale lui Dumnezeu. Într-adevăr, când studiezi biochimia și genetica și life sciences, întotdeauna o să rămâi frapat cât de minunat este Dumnezeu. Unii au înnebunit, s-au crezut mai deștepți ca toți și îl refuză pe Dumnezeu. Și de acolo a ieșit ideea aceasta a evoluționismului. Însă vreau să vă spun un lucru clar. Darwin și evoluționismul au fost învinși de ceea ce este în celulă. Celula l-a învins pe Darwin. Celula l-a desființat pe Darwin. Celula a desființat evoluționismul. Și de aceea o să privim în celulă, pentru că acolo sunt informații extraordinare, care nu pot fi explicate dacă lumea ar fi evoluat pas cu pas. Așa că sper să aveți răbdare cu mine. O să fie vreo oră, e deja 7:10, aveți răbdare. Ok? Încercăm. Însă mesajul are o anumită structură. Go ahead, Alex. So, in the beginning, God created the heavens and the earth. These are words of God from the Bible. And these are words for you, for you, for you, and for me. And the Bible says that all things were made through Him, and without Him nothing was made that was made. Also it says in the Bible, for by Him all things were created that are in heaven and that are on earth, visible and invisible. All things were created through Him and for Him. Last time we looked at a few things and we saw that you cannot be a Christian and believe in evolution. Jesus Christ Himself said that God created men and women according to His own image. So Jesus Christ said God created. And in the Bible it says so clearly that God created the world. So you cannot be a Christian and yet believe in evolution. And if you do believe in evolution, if all there is only a materialistic world, then you are an accident, and life is a blind race without any sense, without a moral direction, and without an intelligent purpose. True, factual science supports Christianity and creationism. There is no conflict. Science does not conflict with religion. Science does not conflict with creationism. Arthur Keith, one evolutionist, said that evolution, and what he said is true, is unproved and unprovable. We believe it only because the only alternative is special creation, and that is unthinkable. And that's the philosophy that we encounter in schools today. We do not want to believe in anything else but evolution, because not doing so means that we acknowledge a God, and we don't want to do that. Evolutionists cannot explain life on Earth, and that's why we should not pay attention to what they are saying. And like I said last time, there is a much greater chasm, prapastia, between the material world and the first cell than the chasm, the prapastia between the first cell and man. It's impossible to have a cell come by itself than to have a cell so-called evolve all the way to a man. When you ask them, can you please explain to me, and we'll see tonight a few things that cannot be explained. When you ask them, sir, Dr. So-and-so, biology teacher or so, can you please explain to me how this could have happened by step-by-step evolution? I guarantee he will say, I don't know, or he will say, somebody else knows, and I'm going to try to find out who. And he will not come back to you with somebody who knows. Because, as we see, evolution means the initial formation of unknown organisms from unknown chemicals produced in an atmosphere or ocean of unknown composition under unknown conditions, which organisms have then climbed an unknown evolutionary ladder by an unknown process, leaving unknown evidence behind them. Okay? But creationism teaches us that God is an intelligent creator, omnipotent, and not bound by space and time. He created everything from His own materials with design and purpose, and each organism was made complete in form and function. Also, each animal that God created, each organism, was created fully functional and was created according to its kind, with the ability to adapt. So yes, we have big dogs and we have small dogs. We have big cats, we have small cats, we have hairy cats and bald cats. But they are all cats. And when I say cats, I include the tiger, I include the lion, I include the cats at their house. Evolution teaches us that from one cell, an organism arose, and from that, a little animal, and from those first animals, then branches diverged, and that's how we have today horses and whales and so forth. Creation, the creation model teaches us that God created each animal according to its kind. And yes, an intelligent creator will allow these animals to adapt to certain conditions, to have more hair, to have less hair, to have bigger muscles, smaller muscles, and so forth. In the DNA, there is room for some adaptation, but they will never jump from one kind to another. We will never see a horse becoming an elephant or a frog becoming a fish. And we saw that Louis Pasteur, by a scientific method, showed and proved that life can only arise from life. Life cannot arise from non-living matter. And with his experiment, he refuted or defeated the law of abiogenesis. That's the basis for evolution, where people say that life can arise from dead matter. Well, Pasteur showed us that such a thing cannot happen. One of the basic, and that's why I put this experiment last time, one of the basic experiments that evolution is being based on is the Miller-Urey experiment, in which in a little flask they put some gases, water, carbon monoxide, hydrogen cyanide, methane, ammonia, and other things, and he zapped them at 60,000 volts for a whole week, and then as soon as something would form in this flask, they would cool it down, and those compounds would accumulate at the bottom of the flask. He had to eliminate those compounds immediately, otherwise they would be degraded by the conditions that were there. And we saw a few things that proved that this experiment, and they did obtain a few amino acids and a bunch of junk, but that does not prove anything. It's as if I take a piece of cement, cement, mix it with water, throw it, and I get a few pebbles, and I say, hey, I got a few pebbles, therefore, that castle formed by itself. I'm sorry, but that castle there, with all its pipes, with all its wires, with all its windows and doors and all the bricks in them, it's nothing compared to what happens in the cell. It's so amazing what the cell has in it. Some counter-arguments to the Miller experiment. We saw that the compounds that he used, and the conditions that he used, and the concentration of compounds that he used, he selected them a priori exactly to fit the composition of the chemicals that he wanted to obtain. There is absolutely no evidence that the atmosphere had any such compounds in older times. Then a methane ammonia atmosphere was impossible. There is no evidence for that to have been present on Earth. And all those chemicals, like I said, cyanide, hydrogen cyanide, carbon monoxide, ammonia, they are highly toxic to life. And you're trying to make life with some toxic compounds. It does not work like that. Also, UV light and the cosmic radiation would destroy and break down ammonia. So ammonia could not have been in those concentrations in the atmosphere. And then the compounds that he produced, they would have to be isolated immediately. Otherwise, they would be degraded. All those amino acids, those few amino acids that he obtained would have been degraded if they would not have been cooled down immediately. And in some books, like I said, they said that life, if it ever rose, it must have arose somewhere under the ocean, maybe under a layer of ice, maybe under 0 degrees. And excuse me, life doesn't happen like that. You don't get life to form at -0 degrees when the atmosphere outside is 80 degrees Celsius. No such mechanisms exist to immediately isolate products when they are formed by thunderstorms and lightning and stuff like that. We saw that oceans, that's what they say, oceans became thick as a bouillon soup. And no evidence for this is in the geological strata. You should see, in the geological strata of the world, you should see layers, or at least one thin little layer of organic matter. There is nothing like that across the entire Earth. And just think about it. If the oceans became thick as a bouillon, ca o supă de roșii, don't you think that extra matter should have been in the atmosphere where the whole thing came? That would have been a very thick atmosphere. And when it filled the ocean would have risen, the level of the ocean would have risen and would have flooded everything. Maybe that's what, it's in their heads. Maybe that's what happened when you put the water and potatoes in a bowl. If you put too many potatoes, the water rises and flows over. But that never happened. Also proteins, because that's what they are trying to make from amino acids to make proteins, cannot be produced in excess water. In an ocean, in a watery environment, you cannot have proteins and amino acids formed because hydrolysis will break them down. Hydrolysis is the combination with water. Also, all organic molecules would spontaneously degrade in time and would not last for millions of years like they need. In fact, millions of years are not needed for anything, because millions of years and oceans of stuff do not matter when I need to have this little compound and this little compound right here and right now. If I do not have them here and now in this very little space here at this same moment, I don't care about the millions and billions of years. I need to have, for a reaction to happen, I need to have the compounds here and now in the proper conditions. And the Miller-Urey experiment allowed absolutely no oxygen, because oxygen degrades proteins, oxidizes them, and they didn't want that. Water. If you have water, water will undergo electrolysis, which means breaking down of water into hydrogen and oxygen, just due to the UV light from the sun and cosmic radiation. So the moment you have water, you will have oxygen and you will have hydrogen if there is a sun shining outside. So also there are lots of layers in the geological strata of iron oxides and other metal oxides, and all these prove that oxygen was always present. And if you do not have oxygen, guys, this is a good one. Listen, if you don't have oxygen, then you don't have ozone, which is made out of oxygen. And if you don't have ozone, any life that would have formed would have been destroyed by the UV light from the sun and the cosmic radiation. So the Miller-Urey experiment did not prove anything, but it actually disproved the law of biogenesis. And also we looked at proteins that are made out of amino acids, and all the amino acids are always in two different forms, depending on how they shift the polarized light that shines on them. You have them in the D form and the L form. They are the same, but they have the same elements in them, but they are not the same. You cannot superimpose them. And anytime you have a synthesis in the laboratory to make amino acids, you will always have a 50-50 mixture of L and D forms. Well, guess what? Life, by God's grace, is all made of L amino acids. All the amino acids in all the proteins, in all the bodies, in all the world, are all just L amino acids. So we looked last time at the probability of a simple protein forming by chance just from L amino acids. And we took a coin and we flipped it, and if you flip a coin, you have 1 in 2 chances of getting tails, right? If you want to have tails twice, you have 1 in 4 chances. If you want to have tails 3 times, you have 1 in 8 chances. If you want to have 100 times in a row tails, you'll have 1 in 2 to the 100th power chances of getting tails 100 times, which is 1 chance in 10 to the 30th power. But the simplest living cells would need to have at least 124 proteins, the minimum. No organism exists like that, but that's the minimum. At least 124 essential proteins for life to occur, each of them with at least 400 amino acids. Well, the chance of having one amino acid with one protein with 400 amino acids in L form is 1 to to the 1 in 2 to the 400th power, which is 1 in 10 to the 120th power. So, how about if you have 124 proteins, you multiply that, and you get 1 in 10 to the 14,880th power. That number does not exist. That number is just on paper. It's utterly impossible to have anything like that happening by chance. And if chance guided the formation of proteins, then the law of probability forbids such an event to have happened. And if the probability of things happening by chance is 0, then the probability of creation is 100%, because we only have these two options, either evolution or God's creation. And if evolution is a 1 chance in 10 followed by 14,000 zeros, that's 0. That is 0. That doesn't happen. Therefore, then, the chance of things happening by creation is 100%. To believe in evolution is to violate the law of biogenesis, the law of probability, and many other laws of physics and biochemistry. And now we come to today's lesson. I know it's long. This review, I know it was a little bit long because I wanted to say a few more things, and the youth care au lipsit și au mers la Super Castron, la Super Bowl, I still wanted them to hear this, because they will hear this in school, and they are the ones who are going to be confronted. Dacă astăzi nu înțelegem limba engleză și în seara asta nu înțelegi most anything, I apologize. But most likely, if you don't understand English, you won't be, dacă nu înțelegeți engleză, nu voi o să fiți cei care o să fiți confruntați cu problemele astea. Dar tinerii care merg la școală, ei o să fie confruntați exact cu problemele astea. Ei o să fie cei care o să ia lecțiile astea de biochimie și de biologie și de zoologie și de genetică și de immunologie și toate astea. Și ei o să fie cei care o să fie încercați să fie spălați la creier. Cei care suntem mai în vârstă, noi, prin harul Domnului, deja ne-am câștigat o stabilitate în credință și pe noi nu ne mai pot clătina. Ei încă nu cred. Tinerii noștri, care nu sunt predați Domnului, încă sunt pe o cale care duce la pierzare. Și dacă ei nu-și predau viața Domnului Iisus Hristos, Creatorul lumii, ei vor fi pierduți pe vecie. Și de aceea o să facem un mic sacrificiu ca să îl lăsăm pe Paul să vorbească în engleză, chiar dacă noi nu înțelegem. O să încerc să vă spun și în românește din când în când. Dar astăzi o să vorbim despre DNA. DNA is God's signature in nature. Unde spune aici că dacă te uiți cu băgare o să-L vezi pe Dumnezeu. Ne-am uitat cu băgare de seamă în celulă, in the cell, we looked carefully and we saw God's signature. DNA, I think, is God's signature in nature. Because DNA is so amazing, like you'll see tonight, that it could not be explained otherwise than being made by a wonderful creator. Let's say you go on the beach and you walk along the beach, it's a shiny day, and you stumble across a bottle. And you pick the bottle, and you look inside, there is a piece of paper. You take the cork out, you take the paper out, and you read, I'm doing so fine. I have a big treasure with me. Come and get it. Or some sort of a message. How many of you would not think that message was written actually by a person? How many of you, guys, how many of you would think that piece of paper came by itself? Leave that on. Leave it on. Yeah. L-ai și acolo? Thank you. How many of you would think that paper just came by itself? No. The logical conclusion would be that piece of paper was written by at least a halfway intelligent guy who knew to write and read, you know, and he put it in a bottle and he threw it away. For a second, you would not even consider that piece of paper was not written by a man. Not even 1 second. Guess what? DNA is a language that was written by somebody. And we'll see in a second what that entails. Language is a product of intelligence. You cannot write and not be an intelligent person. If you don't know to write, you can still be an intelligent person. You just didn't learn to write. But if you know to write, that proves that you have a mind. And intelligence is only the product of a mind. Intelligence does not come, inteligența nu apare din non-inteligență. Inteligența și limbajul este o indicație și este o creație a inteligenței și inteligența este produsul unei minți. Sau dacă vezi un scris undeva, să știi că o minte a existat acolo care a scris-o, care a scris lucrul acela. Intelligence is only the product of a mind, and language is information. Information. If you want to communicate, you need to, writing is a form of communication. Information is not something that's derived from matter and from energy. Information is derived only from intelligence. It's not a property of matter. Intelligence is a property of the mind. To communicate, you need a certain set of rules, and I'm thinking about writing, yes? And also, when you look at that writing, you'll see the same words, you'll see the same letters here and there, here and there, here and there, but there needs to be an irregularity. The words are not the same length, the words are not all the same. If you'd find a book that has the word and forever, you wouldn't think that that communicates information, right? There's no information there. It's just and. It makes no sense. Why am I saying that? Some people will say, hey, crystals. Crystals form structural forms. Crystals will form structures if left to themselves. Pack, 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 pack. You have nice crystals. There you go. You have a proof that matter has in itself the ability to organize itself. Guess what? Crystals are repeated sequences of the same and same stuff. Those molecules have the information in them, the properties in them to link to each other in a certain way. But they will never produce information because they will always be same, and you make a big crystal. That is not the proof for the immense amount of information that's in DNA and in life molecules. Okay. Let's look a little bit at the cell. What's going on? Okay. Anyway, a language that's very interesting today is the binary code. The binary code is used in computers. Limbajul binar este folosit în computere. Și the way computerele lucrează, fraților și surorilor, este că avem acolo un întrerupător care îl punem on-off, pornit, oprit. Sau gândiți-vă la un beculeț. Aprins, stins. Și oamenii au făcut a little code, au făcut un cod în care 8 din astea aprins, stins, pot să pun aprins, stins, aprins. Ei, lui ăla îi dăm să fie litera A. Când fac aprins, stins, aprins, ăla e litera B. Și așa funcționează computerele. Și tot așa sunt comenzile care sunt date în computers. The binary code uses zeros and ones. It was a code created by a German Leibniz in 1679, not too long ago. He found out that you can actually write in binary code pretty much a whole lot of things just with zeros and ones. And actually, he said that this is proof that God exists. Zero being nothing, one is God, and from nothing with God, everything can be created. And that's an idea you should think about. But the binary code requires intelligence. any new information that you want the computer to have, or any new function that you want the computer to have, you need to put your little head to work, and you need to write the code, and if you write the code, that computer will perform. Information is the basis also for any form of life. Somebody thought about how to make life, and he created wonderfully. Let's look at the cell. Today we look with a microscope. The cell, it's so small. And people always have known că celulele se divid. Oamenii au văzut că celulele se divid, dar numai nu știau cum. They didn't know how. Și au observat că acolo în celulă este un nucleu. There is a nucleus in the cell. And they saw that some things happen when they looked with the microscope. Some things happen there at a certain moment, adică cromozomii încep să se condenseze, and then celulele se divid. The cells divide at a certain point. And then they knew that the nucleus has something to do with the command for the cell. That's like the brain for the cell. And they did not know what that matter is that produces all these commands. So in 1953, Watson and Crick discovered the structure of the DNA, which is deoxyribonucleic acid. That's where DNA comes from. And I have here a little model that David has made pentru el la școală în clasa 7. Vedeți ce fac copiii în clasa 7 astăzi? Și nu numai el, mai mulți din clasă, așa că nu el buricul pământului, dar mai mulți au făcut experimentul ăsta. Și DNA, asta este structura DNA-ului, care e acid dezoxiribonucleic, în românește ADN. DNA contains all the information needed for the formation and functioning of each cell. Acum, dacă vă uitați aici, acest double helix, care este DNA-ul, e format din două lanțuri care se înfășoară în jurul altuia. Și o să vedem că cei aici, prin modelul ăsta, cei portocaleu, sunt niște sugars, niște zaharuri, riboza, da? În forma, by the way, in DNA, all the sugars are in D form. D! All of them. Isn't that wonderful? Acum Dumnezeu și joacă stânga-dreapta. Amin. Și lanțul ăsta, avem 46 de cromozomi în celula umană. În fiecare celulă aveți 46 din aia, de iepuraș din aia. In every cell you have 46 chromosomes, except in the reproductive cells. And that's another level where you can punch the evolutionists in the nose. How in the world a little group of cells decided to have only half of the genetic information? That's another subject. But everything has 46, in our case, in humans, but a little group there of cells, they only have half because they need to unite mama and daddy, and they make 46 and become something normal. If mama would have 46, daddy 46, next kid would be 92, and the next kid would be 184, and so forth. Poor kids. They would be monsters. So God is wonderful. And look at the structure, this double helix. Noi avem 2 metri de DNA in each cell. We have 2 meters, 2 metri de DNA, de ADN în fiecare celulă. Avem 100 de, we have 100 trillion cells in our body. I have 100 trillion cells in me. Multiply that, and you can go to the moon, from the earth to moon 500,000 times only with the DNA from my body. In one thumb, I have more DNA to go to the moon and back if I put it end to end. This is 2 meters. That's how much DNA you have in each cell. And you know how big of a cell is? Look in your Bible, find a dot, and in that dot, put 1,000 cells. And the nucleus, the DNA is in the nucleus. And the nucleus is not even half the size of the cell. And yet, you can't even see it, and so much happens there. It's a miracle, brothers and sisters. The DNA of 100 billion people, you can fit it jumate de pilulă of aspirin, half a tab of aspirin. But look how wonderful God decided, how am I going to do with these 2 meters in such a small cell? And he created some little proteins. 8 of them make a histone. 8 histones make a little square thing, cubic thing. And the DNA is wrapped around it. And you make a little strand. And that strand is wrapped around, is supercoiled. And then those are supercoiled so that this string will not tangle. Eu făcut așa frumos, eu înfășurat pe măsor pe fiecare de 2 ori și următorul de 2 ori și următorul de 2 ori și după aceea lanțul ăsta l-a pus așa, lanțul ălălalt l-a pus așa, ca aceste informații astea să nu se încălcească. And when the cell divides, we're going to look in a second, those chromosomes are being caught by some fibers there somehow, and after they divide, adică se înmulțesc, se double, se dublează, some fibers pull them apart. And now you have 2 cells. It's amazing. It's amazing. This is the DNA, cum am zis. You have 1 molecule of sugar, which is ribose. And look at that little thingy there, because you're going to see that picture in a second. Okay, sorry. Ribose is that 5-sided thing. So you have the sugars and the phosphate, which make this band. And in the middle, you have the bases. And there are 4 bases. This is the code that God created. Dumnezeu a folosit un cod, nu cu 0-1, ci cu 4 numere. 1, 2, 3, 4. Those are bases. And they are adenine, cytosine, guanine, and thymine. And in RNA, you change thymine for uracil. Acum, 1 phosphate, 1 sugar, and 1 base is called a nucleotide. There are 3 billion nucleotides in every single cell. 3 billion pairs of nucleotides in every single cell in your body. Which means there are 6 billion nucleotides in your body. In every single cell. In every single cell. And if you were to write a letter for each of these, and you would put those 3 billion nucleotides on a piece of paper, on a regular piece of paper, which would contain about 2,000 some letters, you would get a stack of you would use 1.2 million sheets of paper, a stack of 130 meters tall. O carte cu 130 de 130 de metri înălțime, ca să pui toate numele la fiecare literă, la fiecare nucleotidă dintr-o celulă din corpul tău. În fiecare celulă. In all those 100 trillion cells. So isn't that amazing? It's amazing. Dar ce vreau să spun că am început cu limbajul. DNA-ul hides a code, a language of instructions. And those are called the genes. Genes are pieces of DNA which have the information for making a certain protein. So you have this little piece of DNA codes for hemoglobin. This piece of DNA codes for insulin. This piece of DNA codes for keratin in your hair. This piece of DNA codes for collagen in your bones, in your teeth. Or this piece of DNA codes for myosin in your muscles. Deci, genele sunt cele care codifică, au toată informația necesară ca să se producă o anumită proteină. Și proteinele astea le avem peste tot prin organismul nostru, de la hormoni, la coenzime, la mușchi, la dinți, la oase, la piele, la unghii și la ce vreți. Cum am zis data trecută, dacă ai un om de 100 de kilograme, 60 de kilograme sapă și din alea 40 de kilograme, 15 kilograme sunt proteină. Iar restul, grăsime, la unii mai mult sau mai puțin. Deci fiecare genă codifică pentru o anumită proteină. În codul uman, in the human genome there are 30,000 genes, and each gene with small variations, with small, whether all the gene is turned on or off, or only a portion of the gene is turned on, each gene can code to on average, have 3 proteins. Some more, some less. And each nucleotide, each 3 nucleotides, a very smart guy found out that a group of 3 and another group of 3 and another group of 3, each group of 3 nucleotides, which is called a codon, codes for one particular amino acid. And the amino acids link together in chains, and that's how they form the proteins. And then the chain is folded and is made in the structure that will fit exactly the other molecules that it has to deal with. It's something like a key and lock. It's amazing. So DNA codes for amino acids. Each codon, group of three nucleotides, codes for a particular amino acid. Each amino acid comes together and forms a chain, and that's how proteins are formed. They are chains folded in different shapes, like beads on a string. And the proteins are the ones that control all the functions in the cell. Deci proteinele sunt cele care controlează toate funcțiile în celulă. Why DNA could not possibly form by chance? By now, you're smart guys. You should know why they don't form by chance. Because it's impossible. It's too much intelligence. But let's see a few of the arguments that we have that you can use in school when your teacher tells you that, hey, you are a monkey without hair. DNA sugars are all in the D form, 100%. And again, these sugars are formed whenever you synthesize them, they are formed in L and D, 50-50. If you were to change, to have by chance 6 billion of amino acids put in sequence and they're all in the D form, what's the probability of that happening by chance? Let's have 2 hours now to calculate that. No, it's 0. It's 0. That probability is 0. We saw that for hundreds of amino acids, it's impossible to have formed by chance. How about actually 6 billion? Because it's 3 billion pairs of nucleotides. How about 6 billion? What's the probability of happening by chance all in the D form? 0. Deci probabilitatea ca aceste zahăruri de riboză să fie toți în forma D e 0. DNA dissociates in a watery environment. That's why DNA is contained in the nucleus. The nucleus has a membrane. ADN-ul este conținut în nucleu și nucleul are o membrană cu niște pori. Porii aia, găurile alea, o să le vedem, nu sunt doar niște găuri. Ei acolo au control. Ele se deschid, lasă să intre cine trebuie, închid. E fantastic. Trebuie să ai orice proteină care vrea să intre în nucleu. Trebuie să aibă acolo un barcode, un tag. Dacă nu ai barcode-ul ăla, nu poți intra în celulă. Nici nu poți ieși dacă nu ai barcode-ul. E fantastic. Și DNA dissociates in a water environment. DNA could not form in that soup, dragilor. DNA could not form in that prebiotic soup. Okay? And DNA also decomposes in the presence of amino acids, which they say formed in that prebiotic soup. ADN-ul se degradează în prezența aminoacizilor. De aceea aminoacizii sunt sintetizați afară din nucleu și ADN-ul stă în nucleu, protejat de membrana nucleară sau nucleică. Apoi, DNA will be degraded by UV light. That's why we put sunscreen on. DNA will be degraded by heat. DNA will be degraded by cosmic radiation and all that stuff. DNA will not exist in the presence of abundant oxygen. Oxygen makes it degrade. So ADN-ul va fi distrus de lumina solară, va fi distrus de prezența apei, va fi distrus de oxigen și de alte lucruri. ADN-ul nu poate fi format. Și apoi, care îmi place cel mai mult, is a chicken and egg thing. DNA cannot be formed without a host of pre-existing proteins. We'll see tonight in a little movie. Asta o să fie simplificat. That little video clip. We'll see that ADN-ul nu poate fi format fără să fie niște proteine, cum am zis, at least 124, cel puțin 124 de proteine prezente acolo. Dar de ADN-ul codifică pentru proteine. So you cannot have DNA formed without proteins, and proteins are made by DNA. So you cannot have DNA without proteins, and you cannot have proteins without DNA. Which came first, the chicken or the egg? Care a venit primul? Găina sau ou? Găina a venit, așa este, fraților. Și apoi, pentru că for cells to exist, cells, when Darwin came up with the idea of evolution, their thinking was that man is made of phlegm, and there are some fluids flowing through the man, and each cell is just a blob of amorphic stuff. Deci, Darwin credea că celulele sunt doar un balon de something, de ceva acolo. Dacă Darwin ar fi știut cum arată o celulă înăuntru, n-ar fi pus niciodată teoria asta. If Darwin would have known what's in the cell, he would have never proposed his theory. In fact, Darwin himself said that if we ever find and prove that a process, that something in living creatures cannot be made by step-by-step evolution, then my whole theory would crumble. Deci ce a spus Darwin a fost că dacă o să găsim vreun produs sau ceva în organism care nu poate fi explicat printr-o evoluție pas cu pas, gradată, graduală, atunci toată teoria evoluționismului cade. Și vreau să vă spun că teoria evoluționismului cade. O să ne uităm în altă sesiune că nu se poate să treci de la una la alta în cele mai multe situații în cele mai multe pathways, cărări din organism. So, to have a living cells, for cells to exist, not only DNA is needed, but you need RNA, and you need transfer RNA, messenger RNA, ribosomal RNA, and other things. You need mitochondria by the hundred to produce the energy. You need ribosomes, where we will see that amino acid and protein synthesis takes place. You need endoplasmic reticulum, where all those proteins are assembled. You need the Golgi bodies, where all those proteins are shaped and then spilled out in little transport vesicles. You'll see that's amazing. And you need lysosomes, lipid membranes, nucleic membranes, centrioles. You need pH, care e aciditate, pH regulation, carbohydrates, phosphates, coenzymes, carotenoids. You need a lot of things to have life, not just the DNA. And dragii mei, DNA is the ultimate form of miniaturization. DNA contains more information. DNA in one single cell contains more information than all the encyclopedias of the world put together. And DNA can store information in a smaller space than the best supercomputer on the planet today does. ADN-ul stochează informație într-o dimensiune mai mică decât orice supercomputer din lumea asta ar putea să o facă. DNA este miniatura la maxim, miniaturizarea la maxim. Vreau să vă spun că ADN-ul din corpul nostru se poate dubla. Când celulele se divid, trebuie să dublezi tot ADN-ul dintr-o celulă ca să-i dai și la celula următoare. Să aibă și celula următoare ADN-ul. So it happens, all the DNA replication that happens before the cell can divide happens in 38 minutes, ladies and gentlemen. You know what that means? It's over 1,000 bases per second. I mean, you go through that line, 1, 2, 3, A, C, G, T, A, C, T, A, C, G, G, T. 1000 de ele pe secundă. Isn't that amazing? And it came by chance? No, it didn't. Let's see what a very smart guy says. DNA, ăsta e Bill Gates. Bill Gates, one of the Microsoft creators. He knows all about computers. And he said, DNA is very much like a computer program, except that it is more complex than anything we would ever be able to create. ADN-ul, spunea Bill Gates, este ca un program de computer, de calculator, numai că este mult mai complex decât vom putea noi vreodată să producem. Acum, nu vreau să vă sperii. Și, din nou, înțeleg că uneori informația asta trece așa. Nu trebuie să o memorizați. Doar trebuie să stați, să priviți ce țineți minte bine, ce nu țineți minte e ok, dar cu ce să rămâneți este că Dumnezeu e minunat. Și că Dumnezeu a creat lumea asta într-un mod care nu poate fi explicat să fie apărut la întâmplare. Aici o să vă arăt doar o foaie dintr-o carte de biochimie. Cărțile de biochimie sunt așa groase, fraților. Nu fiecare foaie are reacțiile astea, dar zeci și sute și mii de pagini într-un manual. Asta este molecula de riboză. Asta. Da? Și acolo este grupul fosfat. Asta. Și noi trebuie să formăm, pentru că that's what happens in the cell. This is how the cell produces each of those nucleotides. Trebuie să sintetizeze organismul fiecare din nucleotidele alea. Și nu trebuie să vă uitați, să le memorizați. Dați 11 pași, 11 reacții, just to produce inosine monophosphate, IMP, inosинate monophosphate. Și ăsta este un precursor din care mai sunt două reacții care să formeze cytosine monophosphate, care să formeze citozina, încă doi pași să formeze adenina, adenine monophosphate. Astea sunt reacțiile care trebuie să se petreacă pentru ca una din astea să se întâmple. Those are the reactions that we need to have one of those form. Okay? It's 11 steps. At each step, there are enzymes that are needed. For example, here you need glutamine and ATP. ATP is adenosine, nu mai spun, triphosphate. But you need all those things to have that final thing obtained. And none of these things are useful to the cell unless you get here. The cell will not use for anything else. Celula nu o să folosească niciunul din produsii ăștia, n-are nevoie de niciunul din produsii ăștia. Ce are nevoie e de asta. Și din asta se fac multe după aceea. Cum credeți că a apărut asta la întâmplare? Nu am ochelare, dar să spun că un alfa imidazolfoză nu văd bine, că trebuia să memorizez. E un nume așa de interesant. Ăla să fi fost in the organic soup? I doubt it. Să fi fost ăla? Do you think that thing would have been in the prebiotic soup? Just laying around, hey, I'm here, alpha adenosine triphosphate laying around to be picked up in a reaction? It's amazing. God's world is amazing. Lumea lui Dumnezeu este minunată, cum am zis. Și acum o să ne uităm un pic la niște filmulețe. Ei 8. Cred că mai sunt 15 minutes, 12 minutes. Dar asta o să vă placă. Pentru ca celula să se poată diviza, cum am spus, dintr-un bebeluș, dintr-o celulă care nu se vede, e cât un punct. Ouălețul mamei, it's a huge cell. Și vine și de la tata unul micuț, și aia iar o minune în itself, și se creează prima celulă. Și aia începe să se dividă, dar rapid, pac, pac, pac. E o frumusețe să le vezi pe microscop cum se întâmplă. Și se divid, și se divid, și se fac 2, se fac 4, se fac 8, se fac 16, se fac 32 și așa mai departe. Ca să poți să ai diviziunea celulei, you need first to multiply everything in the cell. Not only the DNA, not only the chromosomes. You got to multiply the mitochondria, the lysosomes, the ribosomes, the Golgi apparatus, everything. You need to divide those. And this little film will show us how that division takes place. It's called DNA replication. Asta ne arată cum într-o celulă se dublează cantitatea de ADN înainte ca celulele să se separe.
DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli.
Bacterii.
A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated.
1000 de ori pe secundă.
DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result.
Este minunat. Toate alea se întâmplă în punctulețul ăla care nici nu-l vedeți. E minunea lui Dumnezeu. Acum, din ADN, from DNA, we're trying now to make the proteins, which are useful for life. Let's look how those proteins are made. So now, we just had 2 copies of the DNA. Acum avem 2 copii de ADN. Celulele se pot divide. But what do you do with the DNA? Let's see how they make proteins. Hai să vedem cum din ADN se realizează sinteza proteinelor care sunt nevoie. Și by the way, it's 38 de minute.
Are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into 2 processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
Acei introni care trebuie scoși din gene, până acum se credea că e junk DNA, adică ADN care nu are nicio valoare. When this video was formed. Now they see that all those portions code for how much you need, where does this molecule needs to go. They are all tags, they are all used in the life process. Let's see how fast this happens.
You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
Asta se întâmplă în organismul dumneavoastră. Și după ce am produs strandul ăla de ARN, care e rețeta, am copiat din carte rețeta, acum trebuie să fac din rețetă proteină. Deocamdată aia e doar rețeta.
The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately.
Proteins.
After dissociation, the polypeptide may need to be modified before it is ready to function.
When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome.
Asta se
When
întâmplă
the chain
după
is
ce
finished,
proteină.
it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
Zici că e războiul stelelor. Dar asta se întâmplă în fiecare celulă. Dacă Dumnezeu nu-i minunat, înseamnă că trebuie să fii orb ca să crezi că toate astea au apărut la întâmplare. Don't you think that what we just witnessed is something smart, the product of an intelligent, amazing God? I think it is. What is the source of the information, of all the instructions for DNA and all life molecules? If evolutionists cannot, and they can't, explain where this information comes from, or how it was generated without intelligence, they cannot explain life. And because it is this information that makes the molecules function as in living creatures. Therefore, the evolutionists are not to be trusted. DNA has more info than all the encyclopedias of the world put together. If we would see an encyclopedia come from space and land on Earth, we'd say, wow, there is intelligence outside, extraterrestrial intelligence. But when we see this happening in the cell, in nature, we don't accept that there is an intelligent God who created that. And I'm going to close with this. DNA is information and language that contains the commands of life. It was not physics or chemistry that arranged the complex and precise sequences in the genetic code, but intelligence. DNA could not exist as such an intelligent and utterly amazing language without an intelligent mind behind it, who is God, the intelligent creator and designer of life. But God has chosen the weak things of the world to put to shame the things which are mighty. The little cell to defeat Darwin, and the base things of the world to bring to nothing the things that are, so that no one should boast in his presence. Because what may be known of God is manifest in them, for God has shown it to them. For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead. So that today you are not with us an excuse that you shouldn't believe in God. So God created the world in a wonderful way. The best thing you can do is to trust in him, give your life to him, say no to all the world has to say, and be firm in your belief that God is the creator and go in the world. Tomorrow when you go to school, be confident that no teacher will be able to defend all these things happening by chance. No teacher will be able to say, hey, this is how it happened. May God help us. Amen. Scuze, a fost mai lung. Îmi pare rău.